All right, time to go teach. Hello and welcome to Lipids, the sequel. Again, we're moving through our unit in biochemistry, and we are just continuing right on through with our sequel to the previous episode dealing with lipids. Targets of the day, here we are. Identify the different types of lipids. So last time we looked at the monomers that make up a lipid, fatty acids, and now we're going to be looking at the different types of lipids that exist. Next, describe the function of each type of lipid, because each type is going to perform a different function. And lastly, describe how those types of lipids interact with water, because, again, water, it's basically the majority of every single cell. And that means that every molecule that's inside of those cells, they got to deal with water. So we need to understand how they interact with that water. Purposes. Well, we got three. First purpose, energy storage. That type of lipid is called a triglyceride. Next, to form barriers. That type of lipid is called a phospholipid. And lastly is to send signals, or to be a signaling molecule, and that type of lipid is called a steroid. So we're going to go through each of these so you can see what they look like and how they do what they do. First, the triglycerides. Remember, they're for energy storage. And now we're going to form a simple triglyceride. The first thing that we're going to need is we're going to need a new monomer. So I know I just said fatty acids, that's a monomer for a lipid. Yes, and also this one. This is called glycerol. Glycerol is a 3-carbon molecule that also has 3 oxygens bonded to hydrogens connected to it. These are called hydroxyl groups. Okay? And each of these hydroxyl groups is going to be important for us as we're talking about building our biomacromolecule, building our lipid. Remember the reaction. We have to remove the components that make up water, which means we have to remove hydrogen and oxygen. Next, we're going to take our fatty acid. And here, I didn't feel like actually drawing out the hydrocarbon chain. It also would have taken up too much space. So I just wrote hydrocarbon. This is our carboxyl group right here. We have the carbon double bonded to the oxygen, and then another hydroxyl group right here. That is what makes up a carboxyl. Now the carboxyl and the hydroxyl are going to interact. And that interaction is dehydration synthesis, absolutely. So, moving on. There we go. We just formed a water, and we form a bond in between these two things. And this would be a monoglyceride. That's what it would be. But we aren't going to stop there because we need to get to a triglyceride. Right now, we have one fatty acid attached to our glycerol, which is called a monoglyceride. Well, let's do this again. And here we are going to bring in a fatty acid. And again, the fatty acid is going to lose the hydroxyl. The hydrogen is going to leave that. And then, boom, we have a bond again. So dehydration synthesis occurs. And we don't stop there because we're going for a triglyceride. And there we go. We have now removed 3 water molecules, and we have bonded 3 fatty acids to our glycerol molecule. This is a triglyceride. A triglyceride is 1 glycerol molecule and 3 fatty acids. That's what makes up a triglyceride. And this is our energy storage molecule right here. So that's what makes up the macromolecule. This is our first lipid. It is generally non-polar. Yes, we do have some oxygen bonded to carbon, absolutely. But that's hidden behind a lot of carbon to hydrogen. And since it's hidden behind a lot of carbon to hydrogen, that means that this is pretty much completely non-polar. There's not a whole lot of polar character in this molecule. It is basically non-polar. This would interact with water pretty much the same way that just a hydrocarbon chain would interact with water, which is to say it wouldn't. It would separate out from water very readily because the water would want to interact with other polar molecules, and not interact with the nonpolar molecule that is here. We have a phospholipid. So for the phospholipid, it matches the same basic starting structure of a triglyceride, but we had to remove one of those fatty acid chains, and we are to free up this hydroxyl group again. I'm just going to rotate some things around their single bonds, so that way we are able to look at it a little bit nicer. So that way you can see what's going to happen at this hydroxyl where our new special group is going to attach. It is called a phosphate group. This is a phosphorus element which is attached to 4 oxygen atoms. That's what makes a phosphate group, and this is why we call it a phospholipid. This oxygen can be attached to a variety of different things. This oxygen over here, you'll notice it has a formal charge of being negative because instead of this oxygen forming 2 bonds, it only forms 1. And since this oxygen only forms 1 bond, that means it's going to have a formal negative charge. So this area right here is extremely, extremely polar, whereas this area down here is very much not polar. And so when we draw it, we typically draw it as a polar head and 2 nonpolar tails. That's how we illustrate a phospholipid, is many times this is the polar head and this is the nonpolar fatty acid tails. The term hydrophilic is based upon the fact that water is polar. So anything that interacts with water is what we call water-loving or hydrophilic. On the other hand, we call the tails that are nonpolar, we refer to them as being hydrophobic or water-fearing. This molecule has both characteristics. It has a polar hydrophilic characteristic, and it has the nonpolar hydrophobic characteristic. And that's really special. So we have a special term for that. It's called amphipathic. It is able to interact with both polar things and nonpolar things. It can interact with polar molecules and nonpolar molecules. So what would happen if we had just a whole bunch of these phospholipids? If you had a bunch of them, they would, arrange themselves in very interesting ways. But what's really important is not how they would arrange themselves in the absence of water, like this. What's really important is how would they arrange themselves in the presence of water? And this is what it would be. They would form a membrane. They would form what we call a bilayer, where the hydrophilic heads would face toward the water, and the hydrophobic tails would interact with each other, making this membrane. So the tails would face inward, and the heads would face outward toward the water. The tails would face into the membrane, and the heads would be facing out of the membrane where the water is. So the hydrophilic areas would interact with the water, and the hydrophobic areas would not interact with the water. And yes, that would mean that the water that is inside of this membrane, well, it would be trapped within that membrane. It would not really be able to get out very easily because of that hydrophobic area that's right there. This is how every single cell is arranged. Every cell on this planet has a membrane that's made of phospholipids. And they arrange themselves in this bilayer. Hydrophilic facing out and all the way in. Sandwiched in between is the hydrophobic tails forming our bilayer, basically making a bubble of these phospholipids. And next, and lastly, we have our steroids. And steroids, well, they don't look like the other types of lipids. They don't fall in with that long hydrocarbon tail because the carbon is going to be in a ring structure. Now you'll notice here, I didn't put on any hydrogens, oxygens, I didn't illustrate any of those. And the reason I didn't show the oxygens or the hydrogens is because it varies by what the steroid is. This, though, is the generic steroid structure. Every steroid is going to have this as its base. It's going to have these carbon rings that make up the foundation of the steroid. So if you ever see a ring structure that looks like this, you should immediately think, oh, that is a signaling molecule. And that is a steroid. You should also be thinking that that's a lipid. And again, it's hydrophobic. It's not going to really interact with water well. So in summary, look, there are three main categories of lipids, and each one performs a specific function. Triglycerides, they are used for energy storage. Phospholipids, those are going to be used to form the membranes of every single cell in existence. It works because of that amphipathic character. If the membrane of a cell was polar, water would dissolve it. If the membrane of a cell was nonpolar, then it wouldn't be able to interact with water at all, and that would be a huge problem for all of life. So the fact that water can't dissolve a membrane, but the fact that it can also interact with that membrane is extremely important. And steroids, they are based upon a ring-like structure of carbon atoms, and they are involved in sending signals. Again, vastly nonpolar in nature. That's it for this time. Be awesome. Stay awesome.